This video will show how to combine markers and traits data to perform association genetics. In this example, I'll use data measured on a tomato core collection population where SNPs and phenotypes information is available. From the Analysis page, I click on the Studies Filter button and select the relevant studies. Here I take genotype studies with marker scores and phenotype studies where traits were evaluated. Now I'll pick this analysis that will display p-value plots along the chromosome. Then I can select continuous or ordinal traits and put them in the Y field. Next I go to the Maps tab. This displays relevant genetic maps that contain markers in the studies I've selected, and I'll put it in the second field. The resulting plot shows the p-value curve along this map, such that each panel is a chromosome. Each colored curve represents a different trait. I can click on a panel to zoom in on the chromosome. And this is the chromosome plot, where each data point represents the p-value of a particular marker and phenotype. Phenotype's colors are given on the top legend. If I hover on a data point, I can view the name of the marker and the phenotype. Then I can click on it, and the statistical comparison between the genotype groups is performed for each trait. The statistics are displayed on the bottom. Alternatively, I can select the p-values table option and repeat the same analysis. Now, results are displayed as a table that is sorted by p-value, and it can be downloaded to Excel. Each row represents a significant effect of a marker on a particular phenotype. The marker's linkage group and position on the map are displayed, as well as the p-value. For the next example, I'll select the Markers Bars option. Here I drag a phenotype to the first field. Then I go to the Markers tab. This displays a list of available markers. I can open a list to view the markers within it. Then I can drag the list into the second field. The resulting figure shows the phenotype's mean for each allele of each of the markers. On the right, there is a legend of the alleles' colors. I can point on a bar to see the marker and allele names, and I can click on it to display the comparison among the corresponding alleles. The HCA option performs hierarchical cluster analysis of the varieties based on their scored markers. So again, I put the phenotype and markers in the appropriate fields and click OK. The results are displayed as a dendrogram. Distance among varieties is based on genetic variation and reflect how much they differ in their marker's profile. A variety's color indicates the value of the phenotype. Green is a low value, black is average, and red is high. The dendrogram is also displayed in a Java application, so it can be zoomed in and out. Moreover, a click on any node in the dendrogram will recalculate the analysis on the subset of lines in that cluster. Let's click on this. And this is the focus of the selected cluster. Going back to the previous tab, I can select the bar option. Now a click on a node will display a bars figure. Here, varieties are ordered according to the phenotypes, and those that belong to the selected cluster are highlighted. Similarly, we can set the distribution checkbox. The distribution figure highlights varieties in the selected cluster. PCA performs principal component analysis of the varieties based on their scored markers and phenotypes. This provides a PCA figure where data points represent varieties. The more distance between data points, the more the genetic variation that exists between them.